Hi, everyone. Good job studying hard. Thank you so much. Today, I want to go over the genetics exam study guide. So, this first part of the study guide. First thing we should note is this is in addition to your previous two study guides that you had. So, this is the third part um, that wasn't on your test but is on your exam. Okay. This first part here is from the inheritance lab. And so if you're really comfortable with inheritance lab, you should do fine on this part. Up here at the top, I have the equation that we use for the chi-square and our t-table, our p-table. And so here's our first question. So in peas, yellow seeds are dominant over green seeds. From a cross between two plants, heterozygous for seed color, 5,945 seeds resulted. 4,452 were yellow, 1,472 were green. Does the data fit the predicted phenotypical ratio of 3:1? Explain. Okay, so first I have this table here. I'm just going to plug in what I know, okay, from the question. So what are my phenotypes that I have? I have yellow seeds and green seeds. Okay, and I'm given some data for yellow and green. Since these were the results of an actual cross that actually happened, I know this is what I observed. It's not necessarily what I expected, but it's what I observed. So 4,452 yellow and 1,472 green. I'm going to put the total here. So it said 5,945. And the reason why I want to know the total is because I'm going to use this total to figure out my expected. Okay. All right, well, to figure out my expected, the first thing I need to do is know what my ratio is. Miss Lisa in this example did give it to you as 3:1, okay? But if you weren't given it, you would now do a Punnett square. Okay, so I did the Punnett square here. It said that we have two plants heterozygous. So I'm crossing two plants heterozygous. I see that 3 display the dominant phenotype of yellow, and 1 out of 4 display the recessive. Okay, so I now made my Punnett square. Okay, it said it was a cross between two heterozygous individuals. So I did that Punnett square. I see I have 3 out of 4 that would display the dominant yellow trait, and I would have 1 out of 4 that would display the recessive green. So what I'm going to do is I'm going to take my total that I have, and I'm going to divide it by 4, because I know that 1/4 of this number must be green for my expected. Okay, so I'm going to plug it into my calculator. And actually, Miss Lisa made a mistake because I got a decimal. But, so I'm going to round this to the nearest whole individual. So my expected will be 1,486. 1,486 is basically 1/4 of my total. Okay. And then if I want 3/4, well, I can just multiply that by 3. So I'm going to do 1,486 times 3. Okay, and let's see what I get. I get 4,450. I'm going to round this time up. Okay. Oh, I erased the wrong value. Okay, so this equals 4,459. Okay, and my total should be the same. Okay, the only reason why I wouldn't is if I was rounding, but if you're rounding one up, the other one should be down. It should total the same. It should be your total individuals. Okay, so I hope you understand now how we got observed. This is what we observed in the question. How we got our expected. We took the total and divided it by 4 and then put it into our ratio. Now we're going to do the calculations here. So you do observed minus expected, 4,452 minus 4,459, which is 7. 7 squared is 49. And 49 divided by my expected, 4,459. Okay, I'll plug that into my calculator in a minute. Okay, observed minus expected, 1,472 minus what I expected, 1486, which equals, let's see, 8 plus 6, 14, 14. Be negative 14, but it's okay if it's negative or positive. I think it was negative. Sorry, I did that quickly. But now I'll do 14 squared. The negative will go away anyway. 14 times 14 is 196. 196 divided by my expected 1486. Okay, let me plug these values into my calculator. Okay, so I did get these values. I hope I didn't mess up on my math anywhere because I wasn't really focusing. Let's hope that Miss Lisa did this right. Okay, so now we are going to be calculating these values, but remember now we have to take the sum of them. Okay, so, and I did round these a little bit. So 0.011 plus 0.3, I'm sorry, I don't think I woke up yet. 0.011 plus 0.132. Okay, so I plugged that into a calculator. I got 0.143. No, 0.143 is my p-value. So now I'm like, I have to compare that value to my table, to my probability table. How many degrees of freedom do we have? Well, my phenotype minus 1, okay, is my degree of freedom. So I have two phenotypes. So 2 minus 1 is 1. And I'm going to go to the 0.05, 3.84. My p-value is less than 3.84. And so the data does it fit the predicted ratio of 3 to 1? I'm going to say yes. My chi-square calculations, one second, I'll say yes, the p-value that I calculated using a chi-square test is less than 3.84, and therefore my data supports. I really should have worded this question differently. I should have said, does your data support the null hypothesis of there being no significant difference between the predictive phenotypic ratio and your data? So, and that would have been easier to say, oh yes, I used the chi-square test and my p-value is less than this. And so the data supports this null hypothesis. However, that's not what the question asked, okay? The question asked, does it fit the predictive phenotypic ratio? So I'm going to say yes, because my chi-square test determined that there was no significant difference between the expected 3-to-1 ratio and my data. Actually, I guess that works. Okay, sorry about going in loops there. Okay, yes, because my chi-square test determined that there was no significant difference between my expected 3-to-1 ratio and my data. Okay, let me grab my charger. Okay, let's move on. In peas, yellow seeds are dominant over green seeds. From a cross between a heterozygous pea plant and homozygous recessive plant, 400 seeds resulted. 125 were green and 375 were yellow. The difference here in this question is I'm not giving you the ratio, the expected ratio. So in this question, I would expect you to make a Punnett square and find the ratio yourself. That is the only difference between these two questions. And maybe the numbers are a little bit easier in this one, okay? All right, so let's do it together. Let's try and fill out my data. What did I observe? I observed 400 total seeds 125, I should put my phenotypes. Yellow and green. 125 were green and 375 were yellow. Let's see, what would I have expected in this scenario? Okay. Let's do my Punnett square. In this case, my Punnett square is between a heterozygous pea plant and a homozygous recessive. Okay, so here's my Punnett square between a heterozygous and a homozygous recessive. If I look here, I'm going to expect 2 out of 4 to display the dominant trait of yellow and 2 out of 4 to be the recessive trait of green. So I'm going to expect half and half of my seeds to be green and yellow. So if my total is 400, half of 400, okay, is 200, and I would expect 200 green. Okay. And my totals should be the same for both columns. All right, well, let's do these calculations. Observe 375 minus what I expected, 200, equals 175. And here I have observed 125 minus what I expected equals negative 75. If I square each of these, let's see, 175 times 175, 30,625. Oh my gosh. Let's see. 75 times 75 is 5,625. Okay. Now I take this value and I divide it by my expected, which was 200. And I get 153.125. I'm sorry, and I get 153.125. Okay, and let's do the same thing here. I'm going to take my 5,625 and I'm going to divide it by my expected. 5625 divided by 200. I end up with 28.125. Okay, for my p-value, I need to take the sum. The sum is, so this plus this is 181.25. Wow. Okay, and how many degrees of freedom do I have? My number of phenotypes minus 1. I have 1 degree of freedom. 1 degree of freedom, 0.05 is only 3.8, and my value is 181. So does our data fit the expected phenotypic ratio? No, because according to the chi-square test that I conducted, there is a significant difference between, my data and an expected phenotypic ratio of 1 to 1. Okay, because my expected ratio was 1 green to 1 yellow. Okay. Why sometimes would, I know this is extreme because Miss Lisa made up the data and I wanted it to be really off, but sometimes why would our chi-square test be off? Perhaps there's something else going on. It's a different kind of inheritance than we thought it was. Maybe it's, sex-linked inheritance or something like that. It could be that they're linked traits. So if we're doing 2 traits like yellow and green, or I'm sorry, if we're doing color and shape, remember how we did like yellow, green, and wrinkled and non-wrinkled? So perhaps we wouldn't get what we expected because these 2 traits are linked, genetically linked in real life. Whereas when we were doing our experiments and putting the straws, the alleles, we were modeling that as if these genes were not linked, right? So we would do these kinds of chi-square tests to help us determine if this trait is actually, if these alleles are actually sorting independently of one another, okay? So those would be some reasons why we would not get what we expected from a chi-square test. If we continue on, in Drosophila, which is fruit flies, Drosophila, the gene for eye color A, wing shape B, and body color C are all found on the same chromosome. The following crossover frequencies for these genes were determined by experiments. What genes must be furthest apart on the chromosome? So if we look here, genes that are further apart have a higher likelihood of crossing over happening between them. Okay? So if there's 18% between B and C, this is the highest value we have in this table. So we know that B and C must be the furthest apart. The alleles for which two genes are most likely to be inherited together from the same grandparent mean that it's very unlikely in the formation of gametes that crossing over would occur between them. And if that happens, you're either going to inherit like both alleles pass on both alleles that were from your mother or both alleles that were from your father. So in this case, the two genes that are most likely to be inherited together would be A and C, because there's only a 6% chance of crossing over happening between them. So A and C, and they would also be the closest together on the chromosome. So now if we want to make a map of this chromosome, so I'm going to draw a chromosome. It says here, we kind of want to think about what percentages add to what. Okay, so I can see 12 plus 6 is 18. So I'm going to say that this plus this equals this. That will kind of make sense in a minute. The percentages, you almost want to think of them as units. Okay? So B and C are 18 units apart. And we said that they're my furthest apart because they have the highest percentage between them. So B and C. And exactly how far apart are they? It's going to be 18. You can think of it like units, 18. Now, where is A? Okay, well, between A and B is 12, and between A and C is 6. So I'm going to put it here. Between A and C is 6, and between A and B is 12. And if you notice here, 12 plus 6 equals 18. So you can kind of check your answers, because if all of the distances make sense mathematically, then you did it correctly. So my distance between A and C plus A to B, does it equal C to B? Yes? Okay, then you did it correctly. This is my order. B, A, C, with A being closer to C than to B. Let's do another one. Okay. So this one shows the crossover frequency for some genes on an autosome of organism Y. Construct a chromosome map. It's so hard to draw with this. Okay. When I look here, I notice that 5.5 plus 5.5 equal 11. So I'm going to say that C and S are far apart, and then it looks like this B is in the middle of them. So I'll put C here, and I'm going to put S here, and we said between C and S is 11. And then we have C and B and C and S. Okay, so looks like B is here in the middle, because B is 5.5 away from C, and B is 5.5 away from S. So from here to here is 5.5. It's so hard to read small. And from here to here, from B to C is 5.5. Okay, so far this makes sense. 5.5 plus 5.5 equals 11. So my CBS is fine. Now, this is like an outlier, okay? C and W is 3%, So I'm going to put W here on the outside. And from C to W is 3%. So when I have like a random one, usually it's like on the outside, okay, of my longest one. So I hope that makes sense. I hope you're able to figure this out, that the order of my genes is W, C, B, S. Okay? If you're having trouble with it though, let me know. Okay, there's one more example here. Let's see, this one looks a little more challenging. Let's take a look at it. Here's my chromosome. I'm gonna kind of look for things that add up to each other. So 25 plus 15 is 40. Let's see if they're kind of talking about the same genes here. A, B, A, D, B, D. I'm gonna say A, B, and D are all within 40 of each other. Okay, so I'm gonna put them here. My B and D are my furthest ones. Okay, B and D are my furthest ones. They're 40 apart. Okay, and we thought that A was somewhere in the middle of them. Between A and D is 25 and A and B is 15. Does that equal? That equals 40. A and D, 25. So if I put A here, between A and D is 25, and A and B is 15. Okay, 25 plus 15 is 40. Okay, so these genes, they match up. Okay, now I have to figure out this B and C, 45. B and C is 45. There's kind of two places C can go, right? Because there's no, I don't know, maybe C doesn't tell me how far C is from anything else. So B and C could either be down here, 45% away, or it could be up here, 45% away. Okay. I don't know. What do you guys think? What one's more likely? I guess we don't really know. So either I would accept either answer of C being here. I think there probably is one that's, oh, wait, that's 45 away, right? So 45 away would be here. C, because B to D is 40, so C must be 5 more than that to be 45. 45. I do think that there is a mathematical way, like I want to say that the crossing over rate can't be more than 50% or something. And I feel like if it was up over here, then it would defy like a different rule of probability. But actually, I don't know for sure. I'm going to put it here. Okay, but if you put it up here, 45 away, that would be fine. Okay, 1 second. Okay, so now we are looking at sex-linked traits, okay? Very important. We didn't get to spend a lot of time with it in class, but the meiosis gizmo is technically a sex-linked trait, okay? From this video, it's me teaching a sex-linked trait. So let's really try and focus and get this. Okay, so it says, it's basically a sex-linked trait is when the trait is on a sex chromosome, either the X or the Y. Often for these questions here, they're talking about the X. Okay. So in humans, hemophilia is a sex-linked trait. That means it's going to be on the X chromosome or Y chromosome, but this one is on the X. Females can be normal carriers or have the disease, but males will either have the disease or not. Okay. So let's take a look at this. Hemophilia is a sex-linked, it should say recessive trait. Like recessive, okay? So a normal female with two X's has two alleles that are healthy, normal. The dominant allele, which is healthy, because we said that it's recessive. However, it's possible for a female to be a carrier. What does this mean? It means that they have the hemophilia allele, but they do not express it and it doesn't affect them. They don't have hemophilia. Why do they not have a hemophilia? Because they have another healthy X. They have a healthy X with a healthy allele, and they're able to make all the proteins that they need. However, if in order for a female to have hemophilia, they have to have two hemophilia alleles. Okay, they have to be homozygous for hemophilia. In this case, both of their X's have the allele for hemophilia, so they are not able to make the correct protein. Now, if we compare that to a male, because a male only has one X, if they have a healthy allele, a non-hemophilia allele, then they're normal. But if they have only one hemophilia allele, then they have hemophilia. Okay, so this is similar to what we talked about with the cat's fur. Okay, it's impossible for a male to be a carrier. Why? Because having just one hemophilia allele gives them hemophilia, unless they have some kind of chromosomal mutation. Okay, so show the cross of a man who has hemophilia with a woman who is a carrier. So let's go ahead and make that Punnett square. Okay, so a man who has hemophilia, what would his genotype be? It would be X with hemophilia and a Y. Those are the two sex chromosomes that he can pass on. Okay? And then a woman who is a carrier, okay, she can either pass on her X with hemophilia, or she can pass on her X that is healthy because she's a carrier, she's heterozygous, she has one of each. Now, if I were to complete this Punnett square, I have to do this quickly, only 2 minutes for class. Okay, they have a 25% chance of having a girl who has hemophilia, a 25% chance of having a girl who is a carrier, a 25% chance of having a boy who has hemophilia, and a 25% chance of a boy who is normal. Okay, so that's how you do that cross. I have to go to class, but I hope that from this example you're able to do the rest. Okay, I'll still make a video for the rest.